Hey guys, Miss Cote here, and welcome to another video in the heredity unit. In the last video, we looked at how Gregor Mendel worked out the relationship between genotypes and phenotypes in his pea plants. However, it's not always that simple. In this video, we'll look at different types of inheritance that don't necessarily follow the Mendelian dominant recessive patterns that we looked at earlier. So let's get started. First, let's look at inheritance patterns that only involve one gene. Three situations where inheritance patterns will not follow simple dominant recessive rules are when the alleles are not completely dominant or recessive. This can be either incomplete dominance or codominance. Second, a gene can have more than two alleles. This is called multiple alleles. And third, in some cases, one single gene can produce multiple phenotypes. This is called pleiotropy. Let's start by looking at when the alleles are not simply dominant or recessive. In these cases, they'll produce a phenotype different from the dominant or recessive phenotype. The examples we were talking about in the previous video were examples of complete dominance. There were two phenotypes for one character, a dominant one and a recessive one. And if the individual was heterozygous, had at least one capital letter, then they would show the dominant phenotype. One exception to this rule is incomplete dominance. Here, the phenotype of the heterozygous individuals is not dominant or recessive, but somewhere in between. It's almost like it's a blend of the other two phenotypes. Here in the pea generation, we're starting with a true breeding red flower and a true breeding white flower. Notice the way your book writes the symbols for the genotypes. They use a capital C, and I'm assuming the C stands for color, with superscripts of R for red and W for white. I know in your previous biology class you may have learned to do these crosses with just a capital R and a capital W. Just do what works for you, but be prepared to understand different ways of writing genotypes. Anyway, if you cross these two flowers, you would assume that the F1 generation would be heterozygous and therefore be red. However, since this is incomplete dominance, the F1 generation are all pink. Like I said earlier, if something is inherited incompletely dominant, the heterozygotes will be a mixture or a blend of the two phenotypes. In this example, red and white make pink. For genetics problems, the question will either tell you directly that it's incomplete dominance, or if you are told that the heterozygotes have an intermediate phenotype, then you're probably supposed to understand that it's incomplete dominance. So if we were to cross two of the pink flowers, our F2 generation would look like this. While the Mendelian genotype ratio would still be 1 RR to 2 RW to 1 WW, the phenotypic ratio would not fall into the 3-to-1 ratio we would expect from a heterozygous by heterozygous cross. Here the phenotypic ratio would follow the genotypic ratio and be 1 red, 2 pink, and 1 white flower. Another exception to complete dominance is codominance. Codominance is when, instead of a blend of the two phenotypes, both alleles are expressed to give the heterozygotes a phenotype different from either the dominant or recessive. So looking back at our flower example, instead of red and white make pink flowers, if the trait is codominant, it would be a combination of both red and white pigment within the same flower. Other examples of codominance commonly seen in problems are chicken feather color, where if you cross a white chicken with a black chicken, you would get speckled chickens who have both white and black feathers, or the roan coat color in horses and cows. You would do the crosses in the same way as incomplete dominance, with the R's and the W's or whatever two letters you want to use, but the phenotype would be different. Don't worry, you're not expected to memorize which traits are incomplete and which are codominant. They'll tell you what you need to know within the question itself. Another type of inheritance pattern comes from having more than two alleles for a particular trait. Remember that there are two alleles for a gene. We get one from mom and the other from dad. And we've been looking at having a choice between two letters, whether they are capital or lowercase, or whether they're the R or W in the case of incomplete or codominance. But what happens when you have more than two choices? That is what multiple alleles is all about. You still only receive two alleles, but there are different possible combinations when you have more choices. Probably the most classic example you will find in textbooks for multiple alleles is blood typing, where three possible alleles, A, B, and O, can give a person one of four different phenotypes. So let's see how this works. What these alleles do is to code for a specific carbohydrate chain attached to a protein. We call these glycoproteins. That is found on the outside of your red blood cells. The A allele codes for the A protein, which in this diagram is represented by a triangle shape. The B allele codes for the B protein, which is shown as a circle shape, and the O allele does not code for any protein. Note how the book uses the symbols of the letter I with a superscript. If you were taught just using A, B, and O, you can stick to how you were taught. Just remember that you need to understand what's going on if you see it written this way on a test. Also know that A and B are codominant to each other and that both are dominant over O. So for a genotype, a person would get two of these three alleles, and that combination would give you a specific phenotype, which is your blood group. For people who have blood group A, their genotype would be either AA or AO. So someone could be homozygous or heterozygous for blood type A. For people who have blood group B, their genotype would either be BB or BO. Once again, either homozygous or heterozygous. For people who have blood type AB, they would have an A allele and a B allele. And finally, for people who have blood group O, they would be the most recessive of the bunch and could only have two O alleles. The types of questions you would be asked about blood typing usually involve being told the blood types of two parents and asking what possible blood types the children could have. Sometimes they'll even throw in a mystery about babies getting mixed up in the hospital, and you have to figure out which baby belongs to which set of parents. Here's an example of a blood typing question that I found off of the internet. If you want, pause the video and try this on your own. So to solve the problem, you would have to do three different crosses, one for each parent. For parent set 1, I'm going to assume that they are heterozygous, just to make sure I cover all options. And from the Punnett square, they could have a child with any blood type, so this isn't very helpful. Let's move on to the next set of parents. Parent set 2 have a 50% chance of having a child with type A blood and a 50% chance of having a child with type B blood. Since there is no child with type B blood that's listed, I'm going to say that they're the parents of child X. Parent set 3, and I'm assuming heterozygous for B to cover all options, have a 50% chance of having a child with type B blood and a 50% chance of having a child with type O blood. And again, since there's no child with type B blood, I'm going to say that their child is child Y with a type O blood. So by process of elimination, that leaves child Z with type AB blood to be the child of parent set 1. The last situation involving a single gene is when the gene produces multiple phenotypes. This is a condition called pleiotropy. While there are no Punnett square problems, you could be given a scenario, asked what type of inheritance pattern it could be, and the answer would be pleiotropy. For example, there are certain diseases like cystic fibrosis and sickle cell where patients would present with multiple symptoms, not just one thing. You can read more about these diseases in your book. Now let's look at two situations where a trait is coded for by multiple genes. The two your book talks about are epistasis and polygenic inheritance. Genetics problems involving epistasis is usually about coat or fur color. A classic example that your book uses is with Labrador retrievers. You can have a black lab, a chocolate or brown colored lab, or a yellow lab. The black and brown fur color is determined by one gene. Black is the dominant allele, so chocolate labs would have two little b's. However, there's also a second gene. Here it is represented by the letter E, and I'm assuming E stands for epistasis, but I could be wrong, where having two little e's, the recessive phenotype, produces no color, so fur would not be black or brown. Instead, that's how you get a yellow lab. So in epistasis, one gene can influence the expression of a second gene. You could be asked to perform a cross between two individuals, and with two genes it could end up being a 16-square Punnett square, or you could just calculate probabilities like we did in class. Here you'll have to pay close attention to what phenotype the combination of letters will be produced. If there's at least one capital E, then you would get black or brown fur, depending on the bees. If there are two lowercase e's, then it doesn't matter what the bees are, the fur will automatically be yellow. Also notice that instead of a 9:3 to 3:1 ratio, the ratio changes to a 9:3 to 4 ratio. So that could be another clue that it's not just simple dominant recessive. Polygenic literally means many genes, and for characters that are coded for by multiple genes, there can be a lot of phenotypes. Examples of polygenic inheritance include height or skin color, and hopefully you realize that there are a wide variety of phenotypes for both of these characters. You can think of these phenotypes as being quantitative, where the more capital letters you have, the taller you are, or the darker your skin will be. If a character is polygenic, then they usually don't have you complete a Punnett square, because let's face it, a Punnett square like this would take you forever. They could have you use the rules of probability to calculate certain genotypes. Most of the time, the question involves something conceptual with no calculations. But looking at this Punnett square, you can see how they simplified skin color into 3 genes. It's really way much more than that, and it shows the range of phenotypes that can occur. If we were to graph this data, we would get a bell curve. Very few individuals would have all lowercase letters and would have very light skin, and very few individuals would have all capital letters and have very dark skin. Most individuals would fall somewhere in the middle. And it doesn't matter which letters are capital, just how many total capital letters you have. The last thing I want to mention is that sometimes your phenotype is not just dependent on your genotype. There are instances where environment can also have an influence on your phenotype. This is called a multifactorial trait. A classic textbook example would be hydrangeas, where in addition to the genotype, flower color also depends on the pH and the free aluminum content of the soil. The pink color is due to soil with a basic pH, while the blue color is due to soil with an acidic pH. The aluminum is responsible for the range in the shades. In the blue one, it could go from light to dark, even into purple. So I hope this helps you to understand the various inheritance patterns that are discussed in Chapter 14. Like the last video, to solve a genetics problem, you need to figure out the genotypes of the parents, set up and complete the Punnett square, and then look back at the question and answer what's being asked. And if they're not direct about the type of inheritance pattern, there is probably a clue somewhere in the question to help you out. In any case, keep working on those problems and be a good human.